So last lecture, in part 1 of glycosides, we have studied different chemical tests, okay, and we have gone through the introduction of glycosides. Today's lecture mainly is on anthraquinone glycosides, and if time permits, we will see few of the cardioactive or cardiac glycosides as well. Okay, sharing with you all the PPT of anthraquinone glycosides. Okay. So anthraquinone glycosides, from the name, it contains anthraquinone nucleus. Okay, and glycoside ka to we already know what is a glycoside. We have already, you guys have already seen part 1. Okay, so let us have a brief introduction about what are anthraquinone glycosides. They are major class of glycosides mainly found in the dicot family of plants like Euphorbiaceae, Polygonaceae, Rhamnaceae, Rubiaceae, etc. Now the family Liliaceae from monocots also show presence of C glycosides. Okay, so when we say anthraquinone, generally they are C type of glycosides. Few of them are also under the class of O type of glycosides. So we will be studying around 6 drugs, okay, under this particular class of compounds. Now, the aglycone moiety here will be anthraquinone, anthranol, anthrones, or dimers of anthrones or their derivatives. Now, what are these anthrone, dianthrone? क्या होता है? That is. Let us have a look at that. Okay. So as you can see here, this is the anthraquinone moiety. Okay. Now the reduced form of this, okay, is a anthrone. And anthrone ka oxidized, actually if you see, it's a reversible kind of a mechanism because of resonance. Okay, so that's an anthranol. So anthrones basically are pale yellow substances which are insoluble in alkali and do not show strong fluorescence with them. While if you see anthraноls, they are brownish yellow substances soluble in alkali and they will show strong fluorescence. So the reduced anthraquinones are biologically more active. Anthraquinones, make a note of this. Reduced anthraquinones, that is anthrones, they are biologically more active. Okay. That is therapeutic activity of anthrone is more than that of an anthraquinone. Now, anthraquinones that are present in fresh drugs, they are generally in reduced form, which on long storage can get oxidized and hydrolyzed. Glycosides of reduced derivatives are more active than oxidized glycols. Understand this point. Glycosides of reduced derivatives are more active than the oxidized form of it. So anthrone is more active than an anthraquinone and an anthranol. This is due to the fact that sugars take the glycosides to the site of action and thus they are more active. Now dianthrones are therapeutically more active than anthrones. A dianthrone will have what? It will have 2 nucleus of an anthrone. Di है ना? तो यहाँ पे भी एक carbon carbon bond से there will be an addition of an anthrone moiety. Okay, so it will become a dianthrone. तो डायएंथ्रोन है तो 2 शुगर अटैच्ड हो गए तो ज्यादा एक्टिव होगा एज कंपेयर टू एन एंथ्रोन. आई होप दिस इज क्लियर. Now anthraquinone, they naturally occur in some plants like aloe, senna, rhubarb, cascara, fungi, lichens and insects where it is serves as a basic skeleton for their pigments. So anthraquinones are also present in fungus or insects where they are going to serve as a basic skeleton, okay, for their basic skeleton for their pigment. नेचुरल पिगमेंट मतलब क्या? कलर. तो कलर जो होता है वो किसकी वजह से होता है? एंथ्राक्विनोन. Natural anthraquinone derivatives tend to have laxative effects. These glycosides are characterized by a chemical test known as Bond-Tranger's test and show the property of microsublimation. Most of this are O and C glycosides, like I said earlier. And on hydrolysis, they will give derivatives of 1,8-dihydroxy anthraquinone, anthranol, anthrone, dianthrone, etc. So the common glycols. Now, if you want to understand glycosides, you have to, particularly anthraquinone glycosides. तो ये 5 बेसिक स्ट्रक्चर्स जो है, समझ रहा है? क्राइसोफेनॉल, इमोडीन, फिसियन, एलोइमोडीन, रीन, ये 5 बेसिक स्ट्रक्चर्स पता होने चाहिए. Okay, these 5 basic structures you should be aware of. Now the sugars present usually are what? Arabinose, rhamnose and glucose. Okay. तो ये 5 स्ट्रक्चर्स से ही एंथ्राक्विनोन्स बनेगा, सारा स्ट्रक्चर्स. ओके, वॉट एवर एंथ्राक्विनोन ग्लाइकोसाइड्स वी आर गोइंग टू स्टडी, वो ये 5 स्ट्रक्चर में ही बनेगा. तो अगर मुझे उसका एंथ्रोन बनाना है तो वी जस्ट हैव टू रिड्यूस इट, और डायएंथ्रोन बनाए तो यहाँ पे मैं एक इमोडीन या एक एलोइमोडीन या एक रीन कुछ भी लगा दूंगी. ठीक है, इमोडीन को या रीन को कुछ और एक लगाऊंगी तो वो बनेगा क्या? डायएंथ्रोन. आई होप यू आर गेटिंग इट. Okay, now what are the 6 different drugs that we are going to study under this class of compounds? So these are the 6 different drugs that we are going to study today. I want you all to get a notebook, a pen and sit with it so that you can make a note of this while you're studying. Okay. So the 6 different drugs we are going to cover under this is aloe, cascara, senna, rhubarb, hypericum and cochineal. First drug, aloe. एलोवेरा तो बहुत लोगों ने सुना होगा, और जनरली एलोवेरा हैज अ वेरी गुड योर स्किन लाइटनिंग प्रॉपर्टीज. ओके, या फिर बिकॉज इट एनहांसेस माइक्रो कैपिलरी सर्कुलेशन, जिसकी वजह से ड्यूरिंग इन द बर्न पेशेंट्स आल्सो इट इज यूज्ड मोर. ओके, अपार्ट फ्रॉम दिस वन ऑफ द अदर यूज इज अ लैक्सेटिव. बहुत लोगों को देखा होगा कि सुबह एलोवेरा का जूस पीते हैं उठ के. Why? Because it acts as a very good laxative. Okay. So aloe is basically a dried juice collected by excision from the base of the leaves of various species of aloe, like aloe perry, aloe vera, aloe barbadensis, aloe ferox, aloe africana, and aloe spicata, belonging to family Liliaceae. Okay, so ये socotrine, curacao, cape aloes. Okay, these are nothing but different types of aloes. Now this is basically how they have given these names. Let us see. So a word originated from an Arabic word aloe, meaning a shiny bitter substance. Now among the different species, vera का meaning क्या होता है? True. ठीक है. Ferox means wild. Spicata means with spikes, flowers and spikes. And barbadensis and africana refers to the habitat of the plant. Habitat yani geographical source of the plant. Okay. So you're getting it. So this refers nothing but to the geographical source of the plant. Now, when I have to look at the cultivation and collection, so genus aloe will consist of around 200 species. Okay, let us not go into detail. This is quite readable as well, but I will highlight few important points. So their plants have rosettes and large leaves, succulent leaves. Okay, it prefers acid, neutral or alkaline soils. So basically it can grow in any type of soils, but it needs proper sunlight. It cannot grow in the shade. Okay, seedlings are transferred to pots and they produce offsets. Now what is an offset? अगर एलोवेरा का प्लांट देखा होगा ना, यहाँ पे भी दिख रहा है इनफैक्ट. ओके, ये लीव्स को, they are different kind of leaves, right? So these are known as offsets. Okay, बहुत लोगों के घर पे होगा भी. It grows very fast. So young offsets are planted in the soil after the rainy season, and then in the second year leaves are collected. They are cut near the base, and juice of aloe is present in parenchymatous cells of pericycle. ये important है. Where exactly the juice of aloe is located? It is present in the parenchymatous cells of pericycle, that are mucilage cells. And in a single incision, the mucilage cells will exert pressure on pericycle cells, and the entire juice from the leaves is drained out. Next, let us have a look at different types of aloes and how they are prepared. Okay, so Curaçao या Barbados aloes, Cape aloes, Socotrite, Zanzibar. Question, मैम ये सब पढ़ना जरूरी है क्या? तो my answer would be yes. Okay, you should know the 4 different varieties of aloe and what are the distinguishing tests to differentiate these 4 varieties. Okay, if you read it once also, it will get registered in your mind. Okay, तो जो भी मैंने रेड में लिखा है ना, that is या मार्क किया है, that is for your point to recollect या remember. ओके उसके आजू बाजू का फिर तुम लोग पढ़ो, याद आ जाएगा ऑटोमेटिकली इफ यू रिकलेक्ट वन दिस रेड पॉइंट्स. ओके, सो बारबाडोस या क्यूरेकाव एलोस में क्या है? वेस्ट इंडीज इज द जोग्राफिकल सोर्स एंड वी शेप्ड वेसल इज टेकन. उसके अंदर सारा जूस दे आर गोइंग टू ड्रेन आउट. ओके, दे विल ड्रेन आउट द एंटायर जूस. This juice will be collected, concentrated either by evaporation or more generally by boiling until it becomes the consistency of thick honey. Okay, now these conditions will favor the crystallization of barbaloin. Barbaloin kya hai? It is one of the main constituents of aloe. Okay, because of the presence of which it becomes opaque, and also it is known as hepatic or livery aloe. Now on cooling, then it is poured into gourds, boxes, or other convenient receptacles and it solidifies. What are Cape aloes? So Cape, they are going generally produced into the goat skins. Basically, it is poured, the juice, जो भी juice निकलेगा offset में से, they are going to pour it into the goat skin. Again, same process. Evaporation allowed for about a month when it becomes viscous pasty mass, and then it is packed into cases. Now in European countries, it is dried in wooden pans with hot air till moisture is about 10%. What are socotrine aloes? So leaves of these plants from which Cape aloe is obtained, they are cut off near the stem and then it is arranged in around in a hole in the ground, which a ship skin is spread. Okay, so एक ship का skin होगा, उसको spread करेंगे and उसके अंदर वो juice collect होगा. So when there is sufficient quantity of juice that is drained from the leaves, it is concentrated by heat or iron cauldrons and subsequently it will be poured into boxes or skins in which it solidifies or cooling. Okay, Zanzibar aloes का क्या होता है? Same thing, but here in this the aloe is also known as monkey skin aloe. Okay, this is also known as monkey skin aloe. Preparation is similar to that of a socotrine aloe, and it is packed in skins of carnivorous animals. Okay, not in the ship, but skin, मतलब ship skin में शायद preparation होगा उसका, but it will be packed in carnivorous animals like monkey skin aloe. तो याद सिर्फ इतना ये रखना है इसमें. Cape will be goat skin, socotrine will be sheep skin, Zanzibar is carnivorous animals also known as monkey skin, and Barbados is generally hepatic, also known as hepatic or liver aloe. Okay, these are different color, odor, taste, and texture of different types of aloes. Now, if you take a single offset, this is written as leaf, but I will name it as an offset. तो उसका माइक्रोस्कोपी अगर कट करके देखेंगे तो क्या होगा? तो अंदर से बहुत सारा जूस निकलेगा. So that is what is there is here. It is generally present here in the aloetic juice. Okay. उसके नीचे होगा the vascular bundles. Okay. And since it is a leaf, it will have stomata. Okay. So inside the pericycle, there is the presence of this aloetic juice. वो हमने already देखा, it is present inside the pericycle cells. Okay, and it will also have calcium oxalate crystals. Coming to the active chemical constituents of aloe, okay, it is a mixture of glycosides among which barbelloin, okay, barbelloin is the major glycoside or chief constituent. So chiefly aloeimodine. Aloin is a mixture of glycosides among which barbelloin is the chief constituent. Okay, now aloin if you see this is a glycoside. Okay, जिसमें यहाँ पे एंथ्राक्विनोन पार्ट क्या है एलोइमोडिन एलोइमोडिन है और उसको ग्लूकोज अटैच किया है तो दैट इज बिकमिंग अ एलोइन विच इज अ ग्लाइकोसाइड. ओके, एंथ्राक्विनोन क्या है या अग्लाइकोन पार्ट क्या है उसका एलोइमोडिन. ओके, chemically aloimodine anthrone is a C10 glucoside and is water soluble. Beta barbelloin and isobarbelloin are also present. Other constituents are aloin, racin, amodine, imodine and aloimodine. Now barbelloin is present in all varieties. Okay, so all four different types of varieties that we saw, sub mein barbelloin is present. It is slightly yellow colored, bitter, water soluble, crystalline glycoside. Now when we see isobarbelloin, it is present in curacao and in trace amounts in Cape aloe, and but it is absent in socotrine and Zanzibar aloes. तो ये isobarbelloin जो है ना, इसी की वजह से distinguishing test हम पढ़ेंगे different. ओके, जो आगे की स्लाइड में आई विल शो यू व्हाट आर द डिफरेंट, बट इतना याद रखना है यहाँ पे that the different distinguishing test will depend on this. Okay. Chief constituents of socotrine and Zanzibar aloes are barbelloin and beta barbelloin. Now the drug also contains aloetic acid, homonataloin, chrysopanic acid, coniferyl alcohol and aloisone. Resin of aloe principally contains aloisin, which is a type of C-glucosyl chromome. Aloisin is also responsible for purgative action of aloes. Apart from Bontrager's rather modified Bontrager's test, it also gives borax test and bromine test. So why I said see this if you see it's a C type of glycoside and that is why I mentioned the word modified Bontrager's. Okay. In case of borax test, we take 10 ml of aloe solution and to it add 0.5 grams of borax. So now a green colored fluorescence is produced. This will indicate the presence of aloeimodine anthranol. So this anthranol is going to react with borax and that is how it is going to give this test. Okay. Next is bromine test where 5 ml of solution, aloe solution and add equal amount of bromine solution. So a bulky yellow precipitate is formed due to presence of tetrabromaloin. Okay. Now what are the uses for this? Both aloes and aloin, they are official drugs. The former is mostly used as a purgative by exerting its action mainly on the colon, whereas the latter is generally prepared over the former nowadays. Now, aloes finds its usefulness as an external aid to painful inflammatory manifestations. It constitutes an important ingredient in the compound benzoin tincture or Friar's balsam. Okay, when I say painful inflammatory manifestations, we have seen that generally in case of burn patients also, it works as a very good anti-inflammatory agent. Okay, aloe gel is made from mucilaginous latex of aloe vera, which is frequently employed in the treatment and cure of radiation burns to get immediate relief from itchings and pain. Aloe gel increases the removal of dead tissue due to its alloctin. Okay, alloctin जो present होता है, that is a content which stimulates macrophage production. तो macrophages अगर बढ़ेंगे तो automatically dead cells फटाफट खा जाएंगे वो लोग, and that is how it is going to improve the removal of dead tissue. Okay, macrophage का काम यही है. तो इसीलिए all your beauty cosmetic products, why it has aloe vera? This is the reason. Aloe usually causes gripping and is therefore administered along with carminatives. See, the distinguishing test that I was talking about is all this: nitrous acid, nitric acid, and cupraloin test or Clune's isobarbaloin test. Okay. Now, if you see nitrous acid test, it has a sharp pink color. अगर याद होगा तो ये सब that is because of isobarbaloin. तो अगर सोकोट्राइन और जैंजिबर में तो आइसोबार्बेलोइन एब्सेंट है, तो दिस विल बी नेगेटिव टेस्ट. ओके, nitrous acid and isobarbaloin, दोनों टेस्ट will be negative. Only nitric acid test will give 4 different shades in the solution. तो अगर चारों varieties को differentiate करना है तो nitric acid test is the best one. Okay. Sharp pink or faint pink क्यों? Because concentration of isobarbaloin is better in case of curecoin and it is in trace amounts in case of K-palose. Okay, यहाँ पे भी वही है. Clune's isobarbaloin, you add a drop of saturated solution of copper sulfate immediately followed by sodium chloride. So wine red color will be given by Curacao and faint coloration changing to yellow will be given by K-palose. What are the adulterants or allied species to aloe vera? It is aloe candelabrum, which is also known as natal aloe, which is dull greenish black to dull brown in color. When scraped, it gives a pale grayish green or a yellow powder. Now these contains nataloin, homonataloin and resin with nataloresinotenol. This can be distinguished as it gives test to borax test and produces a deep blue coloration like Jafarabad aloes. It is a weak purgative. Mocha aloes are the other type of aloe which is used as adulterant. Brittle black and glassy aloe with strong odor. Aloe gets adulterated with black catechu, pieces of iron and stone. Alcoholic extract of aloe under UV light will give deep brown color, while black catechu will give black color. Okay, so these are the adulterants. I just read it because there is nothing much to explain when we talk about adulterants to it. Next drug that we see is cascara bark. It is also known as sacred bark or chitum bark. Okay, so cascara, you can see the picture how the bark looks. It is nothing but dried bark of Rhamnus persiana belonging to family Rhamnaceae. Okay. Now the drug mostly occurs in quilled, channeled or incurved of varying lengths and sizes, usually 20 centimeter long and 1 to 4 millimeter thick, smooth or nearly. So externally covered with a grayish white color, which is usually easily removed and frequently marked with spots or patches of adherent lesions. Beneath the surface, it is violet brown, reddish brown or brownish, and internally it is pale yellowish brown and nearly smooth. Now fracture is short and granular in the outer part and fibrous in the phloem. It has no marked odor but a nauseous, bitter taste. Now, the bark is found mainly to contain two major types of anthracene glycosides. Okay, so हम लोग see, if you see, these are the characteristics of a bark. Now let us see what are the two major types of anthracene glycosides. So normally, if you remember, हम लोग ने जब introduction पढ़ा था, that time I had said कि दोनों क्लासेस में ये bark fall होता है, but majorly it gives modified bond triangles. तो why is the answer here? ठीक है, यहाँ पे देखेंगे कि C type of glycosides जो है वो 90% प्रेजेंट होता है इन बार. ओके, whereas O are present only 10%. तो ज्यादा वो टेस्ट पॉजिटिव कौन सा देगा? It will be modified bond triangles. Okay, so it has normal O and aloin like C glycosides, which comprise about 80 to 90% of total glycosides. So the two C glycosides, which are known as barbaloin and deoxybarbaloin or chrysaloin, are given below. Now, cascarosides A and B are primary glycosides of aloin. C and D are primary glycosides of chrysaloin. So A, B, C, D सबका मैंने यहाँ पे configuration भी दिया है. And what is the R group? This group. Okay, how you differentiate? So aloin और crysalloin का structure क्या है वो भी दिया हुआ है. Okay, so if you just make this substitution, you will understand कि ये जो 5 रिंग मैंने स्टार्टिंग में दिखाई थी ना, उसमें से कौन सी रिंग एग्जैक्टली बारबेलॉइन है और कौन सी आइसोबारबेलॉइन है. Okay, this 5 rings I am talking about. Yes. Okay, CH2OH तो alloimodine है. Correct? So this is how you can come to the conclusion for this. Okay. Now, palmidine ABC, pre-amodine. Palmidine ABC क्या है? That structures we are going to see in the next talk. But that is present somewhat in some amount also in cascara. Okay. Apart from glycoside, it will also contain fat, starch, glucose, volatile oil, malic and tannic acids. Now, how is cascara bark, fresh cascara bark contain? It contains anthranol derivatives which have gripping and emetic properties, and after storage for 1 year, anthraноls are oxidized to anthraquinone derivatives and bark loses its irritant properties. Like if you have a look at the microscopy of this bark, it has cork, then it has cortex, then there are sclerides or sclerenchyma layer that is present. Calcium oxalate prisms are present, secondary phloem fibers and crystal sheath is present. Now if you see along from तो normal जो भी bark का microscopy होता है वही microscopy होगा. Okay, but what is different? Cork will have several layers of cells which contain yellowish substance showing purple color with an alkali. Okay, so when you add a sodium hydroxide or any other alkali for that matter, it is going to give you a purple coloration. Okay, crystal fibers are of diagnostic importance in identification of the powder drug. Now, if you have a look at the different chemical tests, it contains C-glycosides, which gives modified Bond-Ranger's test. It gives red color with 5% of potassium hydroxide solution. It is widely used as a gentle laxative, and the bark also has some of the tonic properties in promoting appetite and digestion. Now, what are the adulterants and substitutes for this? So the bark will have Rhamnus californica, Rhamnus phalanx, and Frangula bark are generally used as a substitute for Cascara sagrada bark. However, the former types of bark of Rhamnus species exhibits a more uniform coat of lichens. Now, what is this lichens? Anyone, can you answer it to me? Okay, this is a uniform coat. Okay, you can this basically are kind of growth you can say. Okay, that is there. Okay, lichens along with broader medullary rays when compared to the original drug species. So Rhamnus alnifolia and Rhamnus corsia are also some few of the adulterants to this major drug. Okay. Next, we come to Senna. Now, Senna, if you see, there are two different types. One is Indian Senna and the other is Alexandrian Senna. So, Indian Senna जो है, उसको we also say it as a Tineveli Senna. Okay, we also call it as a Tineveli Senna. Why? Because it grows somewhere in Tamil Nadu in the Tineveli region. So, it is also known as Tineveli Senna. Now, senna leaf is nothing but it consists of dried leaflets of Cassia acutifolia, known as Alexandrian senna, and Cassia angustifolia, commercially known as Tineveli senna. It belongs to family Leguminosae. Now, senna leaves harvesting is done in three different stages. Okay. First, plucking is done when the leaflets are thick, fully grown. After 1 month, second plucking is done, and last plucking is done after 4 to 6 weeks. So normally these plants are uprooted after third plucking of leaves. Now, if you see senosides, which are one of the major constituents of senna, the content of that is maximum when they are fully grown. After collection, leaflets are dried under shade or indoor by spreading them into thin layers. Okay, then they are packed into bales under the hydraulic press. Now, if when you press this, there will be transverse lines. Now, this is absent in case of Alexandrian cell. Okay, because here the collection of leaflets is done with branches when plants are bearing unripe fruits and dried under sunlight. Now, the process of separation is carried out by sifting, where see, if you see senna, it is also present as pods or stalks. They are separated by manually plucked out. Now, further, these leaves are mechanically tossed, okay, through which all the parts are separated. Entire leaves are handpicked from mixed leaves and packed in mates or bales without pressing. Okay. Now, what are the different chemical constituents? Okay, different chemical constituents of this are four senosides A, B, C, and D, which are dimeric glycosides having their glycones composed of either Re or aloimodic molecules. Okay, senoside A, B, C, and D. If you have a look here, okay, these are the different senosides that are present. A and B will account for its purgative properties. Okay. So again, if you see, this is a nothing but a diacronuclear where two glucose molecules are attached. But ये कौन सा test देगा guys? Bond triangles. क्यों? Because यहाँ पे O glycosides है. Okay, you see the glucose is attached with the help of an oxygen. So this is going to give you a bond triangles test positive. Okay. Senosides A is dextrorotatory, also reported that sugar part of this glycoside has transporting function for the glycone up to large intestine and also protective function so that oxidation of glycone to other very less active anthraquinone is prevented. Now, besides relatively small quantities of monomeric glycosides and free anthraquinones are also present in senna pods. It also contains camphorol. Now, there are two different naphthalene glycosides that are reported. One is tinavalin. Naphthalene क्यों? Because यहाँ पे दिख रहा है naphthalene ring है. Very easily you can remember. टिनेवैलिन ग्लूकोसाइड जो है वो टिनेवैली सेना में है या इंडियन सेना में है. And 6-hydroxymucin glycoside is present in your Alexandrian senna. Okay. Microscopy, like any other leaf, it is a very good leaf. It has the same microscopy, but in this case it is a isobilateral leaf. मतलब क्या कि दोनों साइड पेलिसेड cells present है. So that means it's an isobilateral leaf. And if you see here, this is a Warty trichome. दिख रहा है? Unicellular Warty trichome. Okay. And if you see, stomata कौन सा टाइप का है? Parasitic. मतलब क्या? Guard cells are present parallel to each other. So it's a kind of a parasitic stomata. Then there are xylem fibers surrounded by the phloem fibers. Okay. बाकी तो सब same है. But what is the characteristic of this? It's a thick-walled Warty trichome and prismatic crystals of calcium oxalate. Okay. Now when you see the distinct chemical test, bond triangles तो मैंने already बोला. उसके अलावा mucilage of senna will give a distinct red coloration with ruthenium red solution. Okay. What is the distinguishing test between Alexandrian and Indian senna? So when ether extract is hydrolyzed, with methanolic magnesium acetate solution, Alexandrian senna will give pink color in daylight and pale greenish orange in filtered UV light. Whereas Indian senna will give orange color in daylight and yellowish green in filtered UV light. Tinnevelly senna is invariably found to be adulterated with three cheaper varieties of senna, namely Dog, Palthe, and Arabian senna or Bombay senna. Now, dog senna is also known as Cassia ovata. Palthai is also known as Cassia auriculata. And Arabian senna or Bombay senna is also known as Cassia angustifolia wild variety. Okay. अभी क्या डिफरेंस है दोनों तीनों में, क्यों वो अडल्ट्रेंट है, क्यों वो सब्स्टिट्यूट है, दैट लेट अस सी. Dogs are now ovovate leaves with tapering apex and they show papillose cells in lower epidermis, whereas Palthae contains no anthraquinone glycosides. So if it does not have any anthraquinone glycosides, it is considered as an adulterant. When the leaves are boiled with chloral hydrate, it shows crimson color. Okay. And Arabian senna leaflets are brownish green in appearance and more elongated and narrower in shape. Okay. Uses: senna and its branded preparations are usually employed as purgative in habitual constipation. Glycosides are first absorbed in the small intestine canal, after which the glycol portion will get separated and ultimately excreted in the large intestine. So this is how senna is used. Okay. Any marketed preparation you guys know about senna? Okay, you can write in your comment section. I will tell you two. Marketed preparation, एक तो है senna cot, दूसरा है senna soft. These are nothing but different marketed preparations of senna. Released anthraquinones will irritate and stimulate. Okay, so senna works also as an irritant laxative or a stimulant laxative. Dono different books give different classification of senna, but yes, biologically it works as an irritant or stimulant laxative. So basically, it enhances the peristaltic movements, causing bulky and soft excretion of feces. Inherent action of senna is associated with appreciable gripping, and therefore it is generally dispensed along with carminatives so as to counteract the undesired effects. So this is the color, odor, taste. सबका difference क्या है between Alexandrian and Tinnevelly senna is mentioned here. Okay. Next we come to next set of drug, which is nothing but rhubarb. Now, rhubarb, if we see, it consists of dried rhizomes of Ruium palmatum and Ruium babianum belonging to family Polygonaceae. Indian rhubarb is also known as Ruium imodi. Okay. Now, generally, usko और एक synonym है Ravandchi. Okay. It is nothing but rhizome. Understand, it's not a root, it's not a bark, but it's an rhizome. So Indian rhubarb has roots of emodi, whereas Rapontic rhubarb generally हमें यहाँ पे जो देखने मिलता है that is nothing but Rapontic rhubarb. So in India also when we perform microscopy, बहुत बार it is with Rapontic rhubarb. Okay, now Indian will resemble Chinese variety but are lighter in weight, contains anthraquinone and shows a strong deep violet fluorescence in UV light. It contains hydrolyzable tannins but does not contain rapamycin. Whereas rapontic is pinkish in color, shrunken in nature. It lacks the presence of reed imodine or alloimodine, but it contains rapamycin, which is a crystalline glucoside and is a derivative of diphenylethylene. The glycoside has estrogenic activity and it will show blue fluorescence under UV light. Now, different chemical constituents, if we have a look at for rhubarb, mainly are anthraquinone glycoside and the astringent components form a range between 2 to 4.5%. Okay, so anthraquinones with carboxylic acid moiety or without carboxylic acid moiety. ये भी हम लोग, if you guys know the first 5 structures, you will be able to write down this. Then there are anthrones and dianthrones, plus there are heterodianthrones like palmidin A, B and C. यहाँ पे I will explain you what are heterodianthrones. जो मैंने बोला था आइडियली स्टार्टिंग में, अगर एक स्ट्रक्चर रीन का है और दूसरा स्ट्रक्चर इमोडीन का है या एलोइमोडीन और इमोडीन है, तो दैट विल गिव राइज टू अ हेटेरोडायंथ्रोन. दोनों रीन है तो इट इज अ होमोडायंथ्रोन. But when you have two different structures, up and down, that is nothing but a heterodianthrone. Rhubarb, in addition to the above constituents, will also consist of renolic acid, pectin, starch, fat, and calcium oxalate. Now, there are cytotoxic anthraquinone glycosides which have been recently isolated, named as palmitin. Okay. Different chemical test for rhubarb. So, generally, on being treated with ammonia, it is going to give rise to a pink coloration, and with it gives a blood red coloration with 5% of potassium hydroxide. It also gives a positive indication with modified Bond-Ranger's test. Rapontic rhubarb also gives this test positive. What is the use? Mainly used in the form of an ointment in the treatment and cure of chronic eczema, psoriasis, and trichophytosis. Okay, so it is going to act as a potent keratolytic agent. It is employed as a bitter stomachic into treatment of diarrhea, and it is also used as a purgative. Okay, so this is the structure particularly of palmitin A, which you can have a look here. Next, we see hypericum, which is also known as St. John's wort or goatweed. Okay, now hypericum, if you see, it consists of dried aerial parts of the plant Hypericum perforatum belonging to family Hypericaceae, collected during flowering season or shortly before flowering season. Okay, now Hypericum, what are the different chemical constituents? So it contains naphthodianthrones. Why the name? क्यों दिया है नाम naphthodianthrones? So if you have a look, यहाँ पे बीच में naphthalene ring form हो रहा है दोनों एंथ्रोन के बीच में. That is why the name naphtodianthrone, which are nothing but the anthraquinone derivatives itself. Hypericene and pseudohypericene, hypericide, okay, and flavonoids. Now, hypericide here is a flavonoid. If you see the nucleus, it's a flavonoid, okay, with a glucose moiety. So, hypericide is the proper glycoside, whereas hypericene is an aglycol. If you add to it here a glucose molecule, it will turn to a glycoside. Herb also contains raisins, tannins, etc. What is it used as? An antidepressant. So it is prescribed for anxiety and moderate depression. Hypericine has also shown irreversible MAO inhibitory activity, also reported to have antiviral activity against HIV and hepatitis C virus, indicated in neuralgia, fibrositis, sciatica, and menopausal neurosis. And the last drug that we see for today's lecture is nothing but cochineal. Okay. Now, cochineal is nothing but a dried female insect of Dactylopus coccus costa or coccus cacti. Order दिया है यहाँ पे. Why? Because it's an insect, no? So they've also mentioned the order. वहाँ पे हम variety mention करते हैं. यहाँ पे order mention किया है, that is Hemiptera, containing eggs and larvae belonging to family Coccidae. Now, these insects are brushed from the plants with small brooms and they are killed, certain number being left to provide for subsequent crops. First crop of the season usually contains the most coloring matter. Then the insects, ये important है collection इसलिए मैं पढ़ रही हूँ. The insects are killed by plunging in boiling water by stub heat or by exposure to fumes of burning sulfur or charcoal. If heat is used, the insect will change to a purplish black color and are known as black grapes. Okay, so basically it's a very brutal method of killing the insects. Okay, but still this cochineal is used as a dye. Okay, as a natural dye. Okay. So ma'am, ये glycosides में क्यों है? Because the structure or the dye which is responsible, the pigment that is responsible is the glycoside by its chemical nature. Okay. So, small immature insects and larvae will be separated by seeds, which are known as granula or siftings. Now, the major chemical constituent is a carbonic acid, which is a brilliant purple water-soluble coloring matter. It is a C-glycoside anthraquinone derivative. If you see, दिस इज द स्ट्रक्चर. COOH है, नॉर्मल इमोडीन का स्ट्रक्चर है एंड उसको एक ग्लूकोज लगा है, दैट्स इट. तो अगर वो 5 स्ट्रक्चर्स तुम्हें आते हैं ना, ट्रस्ट मी यू नो एनी स्ट्रक्चर ऑफ एन एंथ्राक्विनोन ग्लाइकोसाइड. फिर वो पाल्मिडिन हो, अपना ये सेनोसाइड्स हो, कैस्केरोसाइड्स हो या एलोइन हो, सब everything you will be able to know if you know that first 5 structures that I have presented in this slide. Okay. Now cochineal and carmine, they are used as coloring agents for liquids and solid and as indicators. No carcinogenic properties have been demonstrated. So that is why, that is what I said, that it is used as a natural dye. Okay, it has no carcinogenic properties as such. So here we come to end of anthraquinone glycosides. Okay, now let us have a look. Okay, so before that, before starting with the introduction to cardiac glycosides, okay, let me take up few of the MCQs, okay, for you all. Maybe cardiac glycosides we might start in our next session, but let us have a look at few of the MCQs or discuss them. I will just share the screen of MCQs. One second. Yeah. So I'm sharing the screen. You can write in your comment section the answers for this. If I get enough comments, I will wait for the next question or I will then go dictating and explaining the answer. So on your comment section and start using it. Okay, whatever we have studied. Let me take up the doubts if I can see any in the comment section. Write in your comment. Okay, so let me take up 5 to 10 of the MCQs. Glycosides are condensation products of what? That's your first question. You can write down A, B, C, or D in the answer. Protein plus glycone, sugar plus a glycone, sugar plus protein, or glycone plus fats. Very easy question. I'm not able to get any comments. Waiting for the comments. Okay, so answer to this is sugar plus a glycone. Next, which of the following do not contain anthracene glycosides? So answer to this, anthracene किसमें नहीं है? We saw to the allocascarines है ना? So Indian squill is the answer. Next, strophanthus do not respond to what? We have not seen strophanthus today, but it is under cardiac glycosides. So it will not give which test? It is B, Van Oort's test, because it is specifically for aldehydes. Which among the following is specific test for ketosteroids? So ketosteroid का specific test is C, which is Zimmerman test. Next, pharmacologically active constituent of cochineal is what? This we just now saw as one of the last drugs. Okay. Yes. Whoever is answering, good. You're answering it correctly. I'm waiting for your answer for the next question. Pharmacologically active constituent of cochineal. Yes, correct. It is no, wrong. It is D, that is carminic acid. अभी देखा ना आप लोग ने. Dash can be detected by Keller-Kiliani test. Okay, this I think I will stop because this is what we have not seen today. So basically, it is digitoxos, or the I will say it is of the cardiac glycosides, the deoxy sugar. Okay, it is not D, it is B. That is digitoxos sugar of digitalis glycoside. Okay, so we will stop here, guys. I have discussed few of the MCQs with you all. Okay, even this we can take up. Clune's isoborveloin test for Curacao aloes will show which color. ओके तो ये लास्ट में हमने देखा था क्लून्स क्यूरेकाउ वाज द फर्स्ट कॉलम राइट सो उसके अंदर इज वाइन रेड इज द कलर ओके so we stop for the today's lecture here. I hope you have understood what was taken up in the lecture. Okay, I will see in the next class again on next Wednesday, where we will be taking up cardiac glycosides and saponin glycosides. Okay, this two we will be covering in the next class, and post that one more lecture where we will be discussing entirely the MCQs and biosynthesis of glycosides. Okay, so thank you everyone for your patient listening. Thank you. I will end the meeting here.